Hi, and welcome to today's webinar. I'm Kathy Kennett, Senior Vice President for Clinical Care with Parent Project Muscular Dystrophy. We are excited to be joined today by Capricor Therapeutics, who will discuss the results of their HOPE-1 clinical trial of CAP1002. Just a few housekeeping points before we get started. You'll notice that the audience is all on mute. If you have a question, please type it in the chat box in the lower left corner of your screen. Many of you submitted questions for our speakers prior to this webinar, so we will try to keep all the questions until the end of the presentation, but we might interject with questions along the way if it looks like people are getting a little bit confused. And as always, a copy of this webinar will be archived on PPMD's website for your viewing later. Now it's my pleasure to introduce Dr. Ron Victor and Dr. Linda Marban from Capricor Therapeutics. Linda Marban is President and CEO and co-founder of Capricor. She's been with Capricor since 2005 and became Chief Executive Officer in 2010. She combines her background in research with her business experience to lead Capricor and create the path towards commercialization for its novel therapies. Dr. Marban began her career in academic science, first in Cleveland Clinic Foundation and then at Johns Hopkins University. She received her PhD from Case Western Reserve in cardiac physiology. Dr. Ron Victor is the Director of Cedars-Sinai Center for Hypertension and Associate Director of Clinical Research. He holds the Burns and Allen Chair in Cardiac Research at the Cedars-Sinai Heart Institute and is lead investigator for Capricor's HOPE trial. Dr. Victor has been awarded numerous grants from the NIH and other organizations and has published more than 180 articles and book chapters in peer-reviewed publication, and is also on the editorial boards of American Journal of Hypertension, American Journal of Cardiology, Journal of Hypertension, and Journal of Clinical Hypertension. So we're very excited that both of you have donated your time and your expertise to this webinar. At this time, I'll turn it over to Linda.
Thank you, Kathy. That was terrific. Shown here is our standard forward-looking statement slide. As a public company, we show this slide to remind you that all of our filings are on record with the SEC. I think Kathy did a lovely job doing the introductions today. What I'll do is I'll just say that we are delighted that Ron Victor, with all of his background and experience in Duchenne muscular dystrophy, both in clinical trials and also in basic science research, has been able to work with us in building our clinical programs for the treatment of DMD and in helping us take our very exciting product, CAP1002, into this indication. So I think you will enjoy as much as I do the presentation that he will be giving to you today. Next slide, please. So the purpose of today's presentation is to focus on CAP1002, which is our allogeneic cell therapy product that we are using currently in the treatment of DMD. The results that you're going to be hearing today are the results from HOPE-1, which was a randomized open-label study of 25 boys and young men with Duchenne late stages. 80% of them were non-ambulant, as you'll soon hear in a few moments, where we were really targeting the cardiomyopathy associated with DMD, and it was an intracoronary delivery of cells. Again, you'll be hearing that in a few moments. And you'll also learn a little bit about how the cells work. The cells work by releasing exosomes, so we consider the cells a natural delivery vehicle for releasing exosomes that then mediate the biologic action of the cells, which you'll hear about in a few minutes. Are multi-targeted. I call them the 5 pillars because they're antioxidative, pro-regenerative, and anti-fibrotic, and most importantly, immunomodulatory, which is actually a little bit stronger than anti-inflammatory in that it helps modulate immune responses, so we get good repair as well as suppression of sort of the bad guys in an immune response. We also have a product in our pipeline called the exosomes. It's called CAP2003. It's a later stage product that is going to be potentially available for DMD as a next-gen product. But right now we're focusing on a congenital disorder of the heart called hypoplastic left heart syndrome, and then other potential inflammatory disorders that we are exploring at this time in a preclinical basis. So stay tuned as this product comes to the fore. So with that, we'll talk a little bit about HOPE. I'm going to actually turn over to Ron on the next slide and let him talk to you a little bit about the HOPE-1 clinical trial. At the end, after we've gone through this data, and I'd like you to focus on the fact that this data suggests that there's both cardiac as well as skeletal muscle benefit in the patients that were treated with the cells. We'll talk a little bit about HOPE-2. Which we've just gotten an IND to start that clinical trial, so we'll be starting it very soon. And we'll give you a little bit of a teaser as to what's coming with another webinar to be planned soon after the first of the year to really update the community on the opportunities for that therapeutic. And with that, I will turn it over to Dr. Victor to talk about HOPE-1, the 12-month results.
Linda, thank you very much. Good morning, everyone. This is the talk that I presented at a late-breaking clinical trial session at the American Heart Association just a week ago. So, next slide. So these are my disclosures. I'm involved with a number of forms of muscular dystrophy research projects. Next slide, please. So dystrophin mutations cause Duchenne muscular dystrophy. This is a devastating X-linked muscle wasting disease that produces patchy fibrosis that's progressive in both skeletal muscle and in cardiac muscle. Typically, ambulation is lost in boys between ages 12 to 15, with premature death occurring before age 30, typically because of failure of pulmonary and cardiac muscles. The only proven therapy for Duchenne muscular dystrophy is glucocorticoids, which can prolong ambulation by 2 to 3 years, can temper pulmonary decline in the teenage years, but cause well-known side effects as shown by the sort of cushion-like appearance of this young man on this slide. So Duchenne really is a disease that is in desperate need of a major therapeutic breakthrough. So the CAP-1002 slides that Linda referenced are putative novel therapy for Duchenne, especially for the cardiomyopathy. So as Linda pointed out, these are cardiosphere-derived cells from donated heart muscle, and the mechanism of action is not that they engraft into the host tissue, but rather that they release these extracellular vesicles called exosomes that target a number of the important cells within diseased hearts. These include the endothelial cells that they're important in blood vessels, fibroblasts, which lead to scar tissue, the heart cells themselves, and inflammatory cells. So, next slide, please. So there are multiple investigational strategies now to try to treat Duchenne. And these investigational strategies target one of multiple different pathways involved. The investigational strategies either target dystrophin deficiency itself, so the dystrophin protein is, for all intents and purposes, absent in patients with Duchenne muscular dystrophy. And one of the more prominent investigational strategies is to repair the disease gene to try to start making some dystrophin. And that the idea is to reduce further cell membrane tears and further loss of cardiac and muscle myocytes. The other strategies focus on one of the downstream signaling pathways, and these include calcium overload, nitroso-active stress, excessive inflammation, inefficiency of the mitochondria, the energy-producing powerhouse of the cell, microvascular ischemia, that is blood flow abnormalities, and all these things culminate in scar tissue formation, fibrosis. What's key about the CAP-1002 cells and their exosomes is that these exosomes target multiple of these pathways simultaneously, multiple of these downstream abnormal signaling pathways. And theoretically, that's a major benefit over some of the other investigational strategies in this field. So next slide, please. So this is preclinical work in Dr. Eduardo Marband's lab with very promising preclinical results. So the CDCs, the cardiac-derived cells, cardiosphere-derived cells, were injected into the heart of the standard MDX mouse model of Duchenne. What you see on the left side of the slide is that intracardiac injection that was given twice improved cardiac function, as shown by an increase in the left ventricular ejection fraction. So the bottom line on this slide, on the left-hand slide, shows the untreated muscular dystrophy mouse. There's a progressive decline over time in the ejection fraction. The treated mice with the CDCs, not only does ejection fraction not decline, but actually increases almost into the benchmark shown by the normal mice. Well, although the cells were injected directly into the heart of the mice, the benefit wasn't limited to the heart muscle. There also was an off-target improvement in skeletal muscle because there was improved exercise capacity. So on this, if you will, 6-minute or mouse version of a 6-minute walk test, the mice were able to walk, instead of 200 meters when they were untreated, the muscular dystrophy mouse could now walk 400 meters, with the benchmark being just over 600 meters. And although not shown on the slide, this effect is not due to improved outflow of blood and cardiac output from the heart. It's due directly to an improvement in muscle function per se, which is very exciting. The challenge is clinical translation. And the reason is that so many other investigational strategies that have shown great promise in preclinical studies, in particular the mouse model, have failed in clinical translation. They failed to show any really clear-cut, clinically meaningful benefit when subjected to the rigors of a randomized clinical trial. So next slide, please. So to meet this challenge, we designed and conducted the HOPE-1 Duchenne trial. This is an early phase called Phase 1 and 2A randomized open-label trial of a one-time multi-vessel injection of a total of 75 million cells into the 3 major coronary arteries. So roughly 25 million cells into each of the 3 major coronary arteries in boys and young men with Duchenne. And this is the first time in the Duchenne field that the therapy required a trip to the cardiac catheterization laboratory. This was primarily a safety trial with multiple exploratory efficacy endpoints performed at 3 sites, here at Cedars-Sinai, at Cincinnati Children's, and at University of Florida, Gainesville. So 25 patients with Duchenne were enrolled. Ages were 12 to 22. All the patients were on a stable steroid regimen for 6 or more months. They had to have left ventricular scar and 4 more segments by cardiac MRI. And by cardiac MRI, the left ventricular ejection fraction had to be greater than 35%. These 25 patients were randomly assigned, such that 13 patients were assigned to usual care, standard of care, plus the CAP-1002 cells, or usual care alone. There was no blinded sham control in this early phase trial, with the follow-up at 6 and 12 months, the last being the 12 months being the primary determination of safety and exploratory efficacy endpoints, in particular by cardiac MRI and in skeletal muscle performance by the performance of the upper limb. And there were additional interim safety monitoring visits. Next slide, please. So here are the baseline characteristics of the 2 study groups. And you can see that the 2 groups were relatively well balanced on most characteristics. Because the average age was in the late teens, which is very unusual for a Duchenne trial, the majority of the boys were non-ambulatory at the time of the study. A bit more so in the CAP-1002 group. than in the usual care group. As expected, there was scar tissue in about 1/5 of the heart. The left ventricular ejection fractions averaged just below 50%. And again, the patients in the active treatment group received just under 75 million cells as designed. Next slide, please. So let's start with the safety outcomes. Very important that there were really no red flag safety issues. The CAP-1002 cell infusion into the heart was generally safe and well tolerated. There were serious adverse events observed in both groups, but not directly related, for the most part, to the infusion of cells themselves. So, for example, in the CAP-1002 group, at the end, one patient did have ventricular fibrillation, an arrhythmia of the heart where the heart starts to beat erratically. That was during the diagnostic cardiac catheterization before any of the cells were infused. And this was transient and led to no adverse effect to the patient. The main adverse events were consistent with an intracoronary infusion procedure, but there was atrial fibrillation in 5 of the 13 patients in the active treatment group. During infusion of the cells into the right coronary artery or the circumflex artery, these are the arteries that supply the bottom part of the heart rate slowed. We see this all the time in the cath lab. And then there was transient atrial fibrillation, which resolved spontaneously. Periprocedurally, there also was an increase in cardiac enzymes. All the boys that received the CAP-1002 cells, and at the same time, 2 of the 12 patients that didn't undergo catheterization in the usual care group had an increase in cardiac troponin, which is one of the proteins in the blood which indicates that there's injury to the heart muscle. You need to understand that what we found and others have known before is that the patients continually release these enzymes into the bloodstream, suggesting that there's ongoing stuttering leakage of cardiac enzymes into the bloodstream. And so the increase in the cardiac enzymes on top of that around the procedure all resolved within 2 weeks. There was no chest pain, there was no heart attack, there was no event that went along with that. Next slide, please. So let's now turn to the exploratory efficacy endpoints and begin with reduced cardiac scar size. This is an exemplary image of delayed gadolinium enhancement on cardiac MRI. And so the contrast material shows up as scar as the white, that's a partial rim around the outside of the heart. The sort of donut appearance is the left ventricle, the main pumping chamber of the heart. And what this shows is at baseline, in both the patient in the active treatment group and the patient in the comparison group, that there's scar in the inferior lateral and anterior lateral segments of the left ventricle. In the patient that received the cell therapy, there's a progressive reduction in the intensity of the white signal, so the scar was resolving, but it was unchanged and remained very intense signal and a lot of scar in the usual care patient. The next slide shows the bar graphs, which is the summary data of scar by cardiac MRI. I want to emphasize this is blinded analysis by core labs, and so by what you see is that in blue are the CAP-treated, the usual care group, and in red are the CAP-treated patients. So the scar increased progressively from 6 to 12 months, as expected in the usual care group, but it decreased progressively in the active treatment group. So at 12 months, the actual amount of scar had increased about 5%, in the control group, and it decreased by 7% in the active treatment group, leading to a statistically significant intervention effect. I want to emphasize that decreased scar is counter to the natural history of Duchenne, in which scar in the heart increases progressively over time. So these are very encouraging results. Even more encouraging is that the reduction in scar was accompanied by an improvement in the function of that region of the heart. So there was no overall effect on global ejection fraction, which isn't surprising because we saw that there was regional improvement primarily in the inferior wall and next in the anterior wall. And it's the inferior wall which is the very segment that is affected disproportionately in Duchenne. It's the first part of the heart that's affected, and it's affected most severely. And then the scar progresses to the anterior lateral wall, and the septum is the last part of the heart to be affected. And indeed, what we see is that the improvement that we saw with the cell therapy follows the same pattern as the natural history that we know about from natural history studies in Duchenne. And I want to turn our attention to skeletal muscle because we wanted to see if we could find a parallel to the ancillary benefit in the preclinical mouse studies where intracardiac injection not only benefited the heart but also benefited skeletal muscle performance. Well, here we focused on the performance of the upper limb because in this older age group of Duchenne patients, most of the boys were non-ambulatory, so standard assessments like the 6-minute walk test were not possible in the majority of our participants. Performance of upper limb is a physical therapy assessment that's rather new to the Duchenne field. So this assesses activities of daily living that depend on the shoulder muscles, the forearm muscles, and the hand and finger muscles. And once a patient with Duchenne loses ambulation, the upper limb performance is absolutely critical to quality of life. So for daily activities like using a joystick to drive a scooter, being able to feed oneself, to use a cell phone and a computer, these are all very critical activities. And the performance of upper limb, as you can see here, tests in a very specific way multiple of these activities of daily living. So because of the age of the patients, the shoulder muscles were already severely affected. And so we combined the middle and the distal score, that's the forearm, hand, and fingers together, where the perfect score is a score of 58, the way this works. And so we focused on, so what we see here, these are mean and median values, and it also shows all the individual patient-specific data, which is very important at an initial trial like this. So what you see is that in blue, again, is the usual care group, and red is the active treatment group that receives the cell therapy. And there is overlap in the two groups, and you can see that there is inter-individual variation within the active treatment group. But if you look towards at the right-hand part of the slide, at the 12-month endpoint, in the patients in the usual care group, all the pulse scores are clustered pretty tightly around either no change from baseline or a small decrease, whereas in the active treatment group, about half or more of the patients showed an improvement in upper limb performance, some rather dramatically. So we investigated this further with two post hoc analyses of the subgroup of patients that didn't have perfect scores to begin with, so that there would be some room to show improvement. And once again, the usual care groups in blue and the active treatment group in red. So with the subgroup of patients that had a score of less than 55, leaving some room for improvement, you define a responder first as a 10% or greater improvement from baseline, or an improvement into the maximal possible score of 58. That if you look at week 6, also at the end of week 12, there's about a 50% responder rate in the active treatment group, and there were no responders defined like this in the usual care group, which goes along with the natural history data. And on the next slide, we slice the data somewhat differently to group patients that either didn't change, so they had sustained performance, or actually improved. And there at 12 months, there were 8 of 9 patients in the cell-treated group responded, whereas none of the usual care group responded. So, and this was statistically significant. So I want to make 3 points in closing. First of all, the CAP-1002 cells delivered by intracardiac infusion in patients with Duchenne is generally safe and well tolerated. These early clinical data are entirely consistent with preclinical studies showing that the CAP-1002 cells benefit both cardiac and skeletal muscle in Duchenne. And a third, the exploratory efficacy analyses signal a potential benefit of the cell therapy for patients with advanced. Thus,
sorry, there's background noise. I'm not sure who's.
Yeah, there's a little bit noise. So to sum up,
if you're not talking, can you turn on mute, please?
Yeah, so taken together, these very promising early clinical data indicate that clearly further research is warranted. And I just want here to acknowledge the funding from the California Institute of Regenerative Medicine. I want to thank very much the foundations, Coalition Duchenne, Cure Duchenne, and Parent Project Muscular Dystrophy, who've been so supportive of this endeavor. And then I want to acknowledge all of our investigators, principal investigators. So thank you so much for your attention, and turn this back to Linda, who will go ahead and explain what's coming next. Yeah, so just go ahead and...
Sure, okay, so
Deborah Eschheim will explain about the outline of the next trial.
Absolutely. Thank you, Ron. That was very, very helpful in a very comprehensive review of the outcomes of the first trial. And based on what we learned from that first trial, from the HOPE-1 trial, and a lot of additional work that's been going on in the various preclinical models looking at intravenous peripheral delivery of the CAP-1002 cells, just to explore whether or not, initially when cell therapy as a field was started long ago, the sense was that you needed to get these new cells, these regenerative cells, into the organs that you needed fixed, which is exactly why the original trials were done, infusing the cells directly into the heart. Since that time, what we've learned, obviously based on some of the data that you've now seen in terms of the effect, even if you give the cells in the heart, the effect on the skeletal muscle, we scratched our heads and said, well, that's really terrific. Let's see if maybe we can avoid having to administer the cells directly into the heart, which requires an invasive procedure, and let's see if we can get the same effects by infusing the cells peripherally into into regular intravenous, say in the arm. And in fact, those results have been repeated in terms of the effect that we see from CAP-1002 given intravenously in MDX mice as well as other preclinical models. And so this next trial will be evaluating the cells intravenously, so we can avoid having to do the coronary catheterization and the direct infusion to the heart. So in addition, that provides a couple of extra pluses. One is we are no longer limited in terms of the dosage that you can administer into a coronary artery. Coronary arteries are pretty small, and so there really was a limitation in terms of the dose that you can infuse into each coronary. So we can administer a higher dose, and we can actually repeat dose, give it several times, because we're not going to need to put a catheter into a heart. So, based on all of the efficacy data that you saw, as well as a whole lot of additional preclinical data in terms of where we think the peak effect happens and over what period of time does it tend to sort of decrease, we will be infusing for this next trial just into the vein CAP-1002 every 3 months. Based on, again, the outcomes that you saw, we will be targeting the effect on the PUL, upper limb function, as the primary efficacy endpoint to be evaluated in this next HOPE-2 trial. We'll still be looking at the heart and evaluating the effect on the heart muscle function. We'll still be looking at a host of additional skeletal muscle effects throughout the course of the trial, including respiratory function, et cetera, and a number of other functional assessments to evaluate skeletal muscle. But we're going to be similarly targeting a pretty advanced population, in fact, a population where it's pretty clear that there are fewer opportunities in terms of clinical trials out there, but one that's really quite similar to the original HOPE trial population. And we are planning, as Ron said, because I'm sure that there are a lot of questions about the HOPE-2 trial, which frankly there's not really enough time to get into in this particular call. So we're planning, as Linda said earlier in the call, to roll out a couple of webinars specific to the HOPE-2 trial design. So that'll be coming very early in the new year.
Thanks, Linda. I have a few questions that have come in regarding the HOPE-1 trial. So you stated that there was improved mobility or improved function of some of the areas of the heart, as indicated by decrease in fibrosis in those areas and increase in movement. Can you maybe explain a little bit about how that movement was measured and why it didn't translate into an improved ejection fraction as it did in the mice?
I'll start with that. First, one thing I think it needs to be emphasized is that while this is an early stage trial, this was a one-time injection of cells into the heart. So to see benefits a year later would be the lower limit of what could be expected with repeated injections. So I think that's really important to keep in mind. Duchenne cardiomyopathy is, as far as I know, is unique in that for reasons we don't understand, the scar begins in the bottom part of the heart, and then this patchy scar progresses around the outer part of the heart and then into the septum at the end. So the areas that were most affected improve the most, which makes sense. So the ejection fractions were minimally low at the beginning of the study, and it would take more patients and I think more injections to expect to see a global improvement. Okay, so because the ejection fractions weren't greatly depressed at the beginning of the trial, you wouldn't expect to see great improvements during the trial, right, Ron? Yes, that's part of it, and the other part is the regional patchy nature of the disease, the scar tissue in Duchenne. It's not like some common forms of adult heart disease that we see all the time, where the heart is globally affected. This is much more patchy and regional. So it makes sense that and when we would see the biggest benefit where the heart needs it the most.
Okay. So in those areas where that fibrosis was patchy and the fibrosis decreased a little bit, were you able to see increased mobility of those areas of the heart? Was that subjectively measured? Okay.
It was measured. Okay. Yes, that's the ability of the heart to thicken, and that improved in parallel with the reduced scar. So basically, the interpretation is that non-functioning scar tissue is replaced by healthy contractile muscle tissue in those areas. Mm-hmm.
All right, excellent. Also, do you know if any of the patients were on any other cardiac medications? I realize that they were all on steroids for more than 6 months, but were they on additional cardiac medications, and were those medications significant to your results, or was that analyzed?
Yes, we can certainly provide additional, and we have all that information, and yes, the majority of patients were on either an ACE inhibitor, lisinopril, or losartan. Some were on an aldosterone antagonist, namely spironolactone or eplerenone. And so the idea was this was on top of the absolute current standard of state-of-the-art care. So yes, this was on top of cardiac medications, and they were fairly balanced distribution in the two groups.
Okay, thank you. Linda, could you maybe talk just a bit about pulmonary function, the exploratory assessment of spirometry in this in HOPE-1, and whether or not you included in HOPE-2 just a little?
I believe Linda might have been muted. Can you add her back to the line?
Yeah. 1 second.
Yeah, and while you're doing that, it's Deborah Ashheim again. So I believe the question was about spirometry and how it was measured in HOPE-1, correct?
Yes. Correct, and if that was taken into account when you analyzed your results and whether it will be included in HOPE-2.
Okay, sure. So, yes, spirometry was assessed in the HOPE trial. It was not, however, designed to be standardized across the institutions, and there wasn't a trial-specific protocol for spirometry. Given how nuanced spirometry is, particularly in this patient population, as you all know, not surprisingly, we really didn't see really any difference between the groups. That said, we are going to be measuring it in a standardized fashion in the HOPE-2 trial. And again, it's going to be an exploratory efficacy assessment, in part also because it remains, a pretty challenging functional assessment to really be able to glean a treatment effect.
Okay, thank you very much. I want to remind everybody who's in the audience, if you have questions, please type them in and we'll be happy to answer them. Linda, can you also speak just a bit about the PROM, maybe a brief explanation about what the PROM is, how that correlates with the POL, and if that was included in HOPE-1 and if it will be included in HOPE-2?
Yeah, can you hear me?
Yeah.
Yeah.
Okay, great. So I'm actually going to pass the baton back to Deborah, our Chief Medical Officer, who I think can give a good understanding of the difference between the two and how they compare.
Sure. So the PROM is a patient-reported tool, outcome measure, directed for upper limb function in the Duchenne population. It was developed in part by you, the patients, the families, at PPMD, a number of the other advocacy groups, and it's clearly targets the types of upper limb function that is prioritized, that really is the most important to you as patients and to you as caregivers of your boys and young men with Duchenne. We did not measure the PROM in HOPE-1. We measured the upper limb function, but not the patient-reported measures. We will, however, be measuring it for HOPE-2. And you know, we think that it will really be an invaluable tool to be able to provide essentially your input into the meaningfulness of some of these quantitative changes we may see.
Okay, thanks. There has been a question about the HOPE-2 trial. Do you know how many boys you will enroll, whether it will be 12 months? And I realized that the HOPE-1 was a safety trial, and so there was not a placebo arm. But can you explain why there might be a placebo arm in the HOPE-2?
Yeah, so this is Linda Marban. Thank you very much. We plan on doing a full reveal of what HOPE-2 will be as soon as we get some of our centers up and rolling so that we have the best opportunity to translate the therapy right into patients. But what I can say is sort of a teaser is that it's a randomized, double-blind, placebo-controlled trial. We did that because we want to speed the path to registration as best as we possibly can. FDA really likes when you have a placebo group, and especially when you have something like the performance of the upper limb as a primary endpoint, which could come under the shadow of volitional effort, which we know most of the kids give their full effort all the time anyway, but it allows you to really parse out the difference between volition and therapeutic effect. We're going to be targeting 84 patients between 10 and 12 sites. There will be sites across the United States and specifically Duchenne centers that we're targeting. And Craig McDonald at UC Davis is our national PI, so we're very excited about that. We think with his involvement, we'll definitely get the best and the brightest working with us on this trial.
Thank you so much. Can you maybe explain how the usual care group was assembled for the HOPE-1 trial and where those patients, how they became enrolled since they weren't treatment patients, and what usual care was?
Yeah. So first, I'm just looking back at the previous question, and I forgot to answer whether it would be another 12-month trial. Yes, it will. This time, though, the boys will be getting 4 doses of cells at time 0, 3 months, 6 months, and 9 months, and then we'll be reviewing the data for efficacy at 12 months. We'll be doing a futility analysis along the way, which we'll talk a little bit more about as we enroll the trial, but won't affect how long the trial will run or the potential endpoint. It'll just be to make sure that the trial is proceeding appropriately. So that's the answer to that question. In terms of usual care, typically these are done in a very standard fashion with a randomization protocol, and sort of to put it in a colloquial fashion, it's like flipping a coin. Heads, you're getting cells. Tails, you're getting usual care. Everything else is kept as much as possible the same between the boys and young men. What I will say about HOPE-1, which was interesting, is that even though it was a coin flip, for whatever reason, the boys in the usual care group had better preservation of skeletal muscle function, and so therefore, we were able to see more potential changes in the therapeutic, in the treatment group, in the group that got the cells, than in the usual care. So what we've done in HOPE-2 is we've tried to make the skeletal muscle function abilities part of the inclusion criteria so that everybody is coming in with approximately the same level of function and therefore can be analyzed together in a better way.
Thanks. The boys that were in the HOPE-1 trial, how long will you continue to follow them and collect data on them? And do you have an idea of how long you will continue to follow them?
Deborah?
Yep. So the boys in the HOPE-1 trial have finished the 12-month active follow-up period, and they're now going to be followed once a year by phone follow-up to complete 5 years. Now, that said, the usual care arm will also be offered participation in an open-label extension for the HOPE trial, the first trial, and that part of the protocol is going to be submitted to the FDA just for clearance, although they knew that this was going to be the case, and we've had some good preliminary discussions with them about it. But that's going to be submitted to FDA. Are we in December yet? It's going to be submitted this month, December.
Okay. Are any of the patients that were included in the usual care model or in the HOPE-1 trial eligible to participate in the HOPE-2 trial, potentially?
Yeah, so that is a great question. They are potentially eligible to participate in the HOPE-2 trial. It depends on whether they meet the eligibility criteria around their muscle, the upper limb muscle function, which is really what we're targeting to identify the populations for this next trial. But in terms of their prior exposure to CAP-1002, as long as they haven't had a cell therapy within a year, which definitionally they wouldn't have at this point, they could be eligible, yes.
Okay. Excellent. Well, I think we have covered all of our questions. Is there any information that you would like to give as a summary?
Well, go ahead, Lynn.
No, Ron, we'd love to hear what you have to say. Please go ahead.
Ron, we'll let you go first.
Well, thank you, Linda. As one of the 3 site principal investigators, I just want to thank all the boys and young men and their families for participating in this trial. This was the first trial, as I mentioned, ever to require a visit to the cardiac cath lab, an infusion of cells directly into the arteries that supply the heart with blood. And the outpouring of participation from the community was just wonderful. So from the bottom of my heart, thank you very much.
Thank you so much, Ron.
That's very typical of you and very typical of the commitment that you bring to the patients and their families. I'd like to echo what Ron said. I'd also like to, again, thank Parent Project Muscular Dystrophy Coalition Duchenne and Cure Duchenne for all of the help they've given us in getting to know this community. I think one of the great joys of developing a therapeutic for this disease is to get to know not only the patients but also their families, and we are working hard to see if we can make a difference for them. In terms of what to look for, let's talk very soon as a group regarding HOPE-2. The faster we enroll that trial and get the patients treated, the faster we'll have data. And while the therapeutic action of these cells have been published in over 100 papers, what we can say is that the combination of anti-fibrotic, immunomodulatory, and pro-regenerative meaning that the actual muscle is created because we've kind of cleaned out the dirt of the playing field. We have an opportunity to 3-prong attack this disease, and we also are working hopefully closely with those that are working with dystrophin replacement therapies so that we can not only have new dystrophin healthy cells, but also continue to work on the immune and inflammatory complications of the disorder.
Thank you so much, Linda. Deborah, did you have any final thoughts?
I think that they've really been covered. I want to add my thanks to Ron, to the community, and specifically to the patients and their families. And we're really excited about what we've seen so far, and we're really looking forward to our ongoing development of CAP-1002 for this really, really just challenging and devastating disease.
Thank you so much. Just there's one other question that came in about the extent of this treatment or how long you expect this treatment to last. And it's my understanding that you'll continue to follow the patients that received CAP-1002 in the HOPE-1 trial to see how long these benefits last, and then cross that bridge with HOPE-2 when that comes. Correct?
Yeah, so, what we saw is that we saw the cardiac effects as well as the skeletal muscle effects with one dose of 75 million cells intracoronary persist or appear to persist out to 1 year. We decided, after reviewing a lot of preclinical data, and there's a tremendous body of preclinical data to support this clinical work, that what we wanted to do is provide every opportunity for the cells to continue to work in a fashion in which they don't get sort of diminished over time. And so we're going to be dosing every 3 months, following the patients for a year. We expect the effects to last somewhere between, let's call it 3 months and a year, but with the redosing, we'll be able to sort of measure the sustenance of that effect. And then the goal would be to, over time, see if we can back off of that dosing regimen to less frequently and sustain the effect. But we wanted to give every opportunity in HOPE-2 for success.
Okay. Thank you very much. With that, I'd like to thank everyone who tuned in, everyone who asked questions, everyone who participated. Remember, this will be archived if you'd like to view it later. I'd like to thank all of our partners with Capricor. We hugely appreciate all the efforts that you're putting into our community, all of the dedication and compassion that goes along with your trial, and we will follow you closely, as usual, and we will be in touch for HOPE-2 very soon.
Thank you so much. Thank you so much, all of you.
Thank you so much. This concludes our webinar.